It's very important that you keep brand integrity here. Photosynthesis green, obviously. Okay, so photosynthesis. Literally, we're making stuff, synthesizing, using the energy of light. This can be divided into two phases: the light-dependent reactions, which take place on the thylakoid membrane, and then the light-independent reactions, I will read that next comment in just a moment, which take place in the stroma. Yeah, sure. You can take screenshots. The whole thing's being recorded, so if I outlawed screenshots, that would be a weird decision. The light-dependent reactions, actually, they're called light-dependent because sunlight is directly involved. We are powering this electron transport chain using the energy found in photons of light. Ultimately, the products are going to be ATP, our old friend, and NADPH. Those two coenzymes are energy-rich molecules. ATP stores energy in the phosphate to phosphate to phosphate bonds. NADPH is carrying around the high-energy electrons that the pigment molecules captured all the energy from. So the light-dependent reactions involve the electron transport chain. Okay. There's two electron transport chains we talked about, one in photosynthesis and one in cellular respiration. The photosynthesis one is the light-dependent reactions. The light-independent reactions require ATP and NADPH, and it takes CO2 and it reduces it in order to get something else called G3P. G3P is the final product. The light-independent reactions are also called the Calvin cycle. Okay, so that happens in the stroma. All right, now those are the big kind of overall idea of photosynthesis. Let's get into a little bit more detail. If I have a leaf and I take a little cross-section of that leaf, I would find that there's actually layers of material in there. Just like if I were to take a cross-section of someone's skin, I would find that there are layers of material. The topmost layer is called the cuticle. It's kind of a waxy protective coating. Then you have these kind of empty cells, not a lot of chloroplasts in this next layer of cells. That's the epidermis, just like we have an epidermis. Next, we have these kind of elongated cells, which are faced vertically up, right, with their long ends pointed kind of up here. And this is called the Palisade Mesophyll. Okay. Now, every layer of cells that sunlight has to go through in order to do for the leaf to do photosynthesis, you're getting less and less sunlight because of that cell wall, right? The sunlight has to go through the cell wall in order to get to these chloroplasts. So we have these protective layers, the cuticle and the epidermis. They don't have a lot in them. They're mostly empty, so a lot of sunlight gets through. And then in order to minimize how many cell walls the sunlight has to get through, we arrange these palisade cells in such a way that the maximum amount of sunlight is going to be hitting chloroplasts. So this is where you would find the most chloroplasts in a leaf cell. You would find it in the mesophyll layer, but the mesophyll actually has two sections to it. You have the Palisade Mesophyll, and then you have this other layer which is called the Spongy Mesophyll. The Spongy Mesophyll has lots of air gaps all over the place, and the reason for that is to allow gas to circulate. On the other side, we have another layer, the epidermis, another layer of epidermis here at the end, and more cuticle. But on the underlayer, there's also another thing going on. And that is these are these little pores. They show up on the top layer as well, but there's more of them on the bottom layer. These are called stomata. Okay. And these just allow gas exchange. So one of the things with photosynthesis is that CO2 has to go in, right? The general equation for photosynthesis is 6CO2 plus 6H2O gives us C6H12O6 plus 6O2, and obviously sunlight is in there as well. That's one of the reactants, is the sunlight coming in here. So in order to get the CO2 into the chloroplasts, into the plant cells, and to allow the O2 out of those chloroplasts, out of the plant cells, we have these little openings on the underside of the leaf, because this waxy layer, if it was just solid, it would be essentially a gas-tight mechanism, so the leaf would just sort of not be able to exchange gases. So the stomata allow CO2 in and O2 out. Okay. So we got the spongy mesophyll here connected to the stomata, and that allows all these materials to circulate around, and it gives the gases access to the palisade layer as well. And then on either side of these stomata, we have what are called guard cells, and they can just kind of inflate to prevent, to close up this little pore. I think of the stomata as being the nostrils of a leaf. It's what they use to breathe. Okay, so if we were to go to one of these palisade cells and not my best arrow, and analyze it, we would find that there are a lot of chloroplasts inside. Chloroplasts have two exterior membranes. So they have an inner and an outer exterior membrane, two membranes. Inner and outer. But then inside, we have this space called the stroma. So don't mix up stroma and stoma. Stoma is the singular for stomata. These are the gas exchange pores, whereas stroma is the space inside of a chloroplast. And then in the stroma, we have these stacks of little green pita breads. And these stacks are called grana, or agranum, if you want to do the singular. So this whole stack is a granum. And then each little pita pocket is a thylakoid. The space inside the thylakoid is called the lumen, or the thylakoid space. Different people call it different things. I always call it the lumen because I think that's a better name. I think it sounds cool. All right. On the thylakoid membrane is where we find the light-dependent reactions. The light-dependent reactions involve a series of membrane-bound proteins, right? So we got Ps2 Photosystem 2, that's the first one in this thylakoid membrane. Photosystems are complexes of proteins. They're just protein complexes, so lots and lots of polypeptides kind of folded together here. And inside their structure, they have these pigment molecules. Pigment molecules are like chlorophyll A and chlorophyll B and xanthophyll and carotenoids, all these things. And what they're good at is they absorb sunlight. Or any light, really, as long as it's the correct color, the correct wavelength. Now, these pigment molecules, they absorb that energy, and that energy gets stored up inside their electrons. So their electrons get charged up, and they actually pass all of that energy to one special pair of electrons, the special pair. It's just a single pair of chlorophyll molecules, which are referred to as the special pair, and they just pass all of the energy right there to concentrate that energy so that we can make an excited electron. Okay. When electrons are excited, they're more mobile. They move around. They get passed from place to place to place, and that's where we get an electron transport chain from. So if you're going to take a screenshot of this, take it now because I've run out of space and I'm going to need to draw out the electron transport chain. We have the thylakoid membrane, and it's got a bunch of proteins floating around in it. I'm not going to draw all the phospholipids, but this is photosystem 2. And photosystem 2, as we just saw, absorbs energy from sunlight, and it charges up an electron, which is then allowed to move down the line. It gets passed to a coenzyme. That coenzyme, I showed you some of them in the slides. We're not going to worry about them too much right now. The next important protein that it gets passed to is Cytochrome B6f. Let's draw it in a kind of elaborate way, I guess. Cytochrome B6f, and cytochrome B6f passes that electron down the line. Now, essentially what we're doing here when we pass an electron from one enzyme to another enzyme to another enzyme, we're running an electric current through cytochrome B6f. So it can do its job under those conditions. A lot of times enzymes are like engines or motors, and if you run this little electric current through them, you can get them to do some work along the way. And the job of cytochrome B6f is to take hydrogen ions and pump them into the lumen. So cytochrome B6f fills up the thylakoid, the interior space of the thylakoid. It fills it up with positive charges every single time an electron is passed by. So we need a steady source of electrons in order to keep this thing going. Photosystem 2 just lost an electron, so it's got a lot of positive charge in it already, and that means it's going to be very greedy to steal some electrons from somewhere else to replace that. And where it steals it from is water. So H2O molecules, the electrons get swiped away, and you end up with O2. We have 2 H2O molecules. You get an O2 and 4 hydrogen cations, which, if they are outside of the lumen, they'll be pumped in pretty shortly by cytochrome B6f. So they all end up in this positive area here. Okay, we just made the electron do some work, so it's lost some energy in that process. The next protein complex it gets passed to is photosystem 1. Photosystem 1 is the second photosystem. I know that's confusing, but I didn't name them. And it works more or less the same. One big difference, like there are some very tiny differences between them. One big difference is that you don't have photolysis. So this process of splitting water molecules and getting oxygen is photolysis. And remember the general equation for photosynthesis, right? You got 6 CO2 plus 6 H2O, light, gives you C6H12O6 plus 6O2. Well, here we're already seeing some of those components come in. Sunlight, light, is being absorbed by the pigments in these photosystems. That's where you would find the most pigments, is in the thylakoid membrane here, right? And they're using that energy to split water molecules and produce O2. So that's photolysis right there. It's kind of hidden in this equation. Right? This equation makes it look like it's one big reaction, but the first thing that happens in photosynthesis is that water molecules are split to make oxygen molecules, and then later we get the CO2 being reduced into glucose. Okay, so the photosystem 1 recharges that electron, so it can continue on its merry way. Photosystem 1 does not need to steal any electrons from water because it has a steady supply of new electrons coming in from further down the line. So it doesn't do photolysis. Photolysis only happens at photosystem 2. The electron is then passed to another coenzyme, but again, I'm going to skip over the kind of mobile electron-carrying coenzymes, the little steps on this process, and just focus on the big ones. We have NADP reductase. Now, enzymes are named after what they do. It's an ase, an enzyme. A reductase, it's an enzyme that reduces things. What does it reduce? It reduces this coenzyme NADP+. And when you reduce something, it's gaining an electron, right? Reduction is gaining, oil rig. So that electron is landing eventually on this NADP+, which then causes it to become NADPH. This is the high energy version of that coenzyme, and it's one of the products that we're making in the light-dependent reactions. Now, it's not the only product that we're making in the light-dependent reactions. We also want to make ATP. In order to, I forgot how PowerPoint works with drawing. Leave a little gap for myself. In order to make ATP, we're going to use chemiosmosis. You guys remember that word, chemiosmosis? Chemiosmosis is the flow of these hydrogen ions that are inside the thylakoid, inside the lumen, out of the lumen into the stroma. And they do that through an enzyme. You guys are going to be saying this along with me at this point, right? ATP synthase. Okay, so these positive ions, they're allowed to flow through, down their electrochemical gradient, flow out. That process of flow is called chemiosmosis, and the enzyme that they're flowing through is ATP synthase. And every little proton, every little hydrogen ion that comes through ATP synthase, we take one ADP molecule, low energy coenzyme, plus an inorganic phosphate that's just kind of floating around out here, and we make ATP. Okay. So we use the energy, we capture some energy from chemiosmosis, and we put that energy into ATP. Now we have our two products of the light-dependent reactions, ATP and NADPH. We also made some oxygen, which is just allowed to leave the plant cells because the plant doesn't really need oxygen right now. It'll need it later if it wants to, do cellular respiration and break down these sugars, but there's plenty of oxygen in the atmosphere. It doesn't need to store it or anything like that. So that just gets emitted out of the plant. So these are the light-dependent reactions. After the light-dependent reactions, we have the Calvin cycle, also called the light-independent reactions. which is the light independent reactions. Okay, so we saw what happens to water in the in photosynthesis. It gets split in order to make oxygen during photolysis. But now we need the carbon dioxide to come in. The Calvin cycle takes CO2 from the atmosphere, and it's going to turn it into a product which we call G3P. So G3P is ultimately what comes out. CO2 enters, G3P comes out. CO2 is a one carbon molecule. It's got one carbon and two oxygens, but we're just counting carbons right now. So in order to make a G3P, we're going to need 3 CO2. There's 3 steps of the Calvin cycle. The first one is called carbon fixation. Step 2 is called reduction. And step 3 is called RUBP, regeneration. Okay, so in the first step, carbon fixation, we have an important enzyme that you guys are going to want to know. Its name is Rubisco. Dang, Ed, you thirsty? Rubisco takes CO2 and another kind of coenzyme around here called RUBP, and it locks them together. Here's my Rubisco. Locks them together in order to make a 6-carbon intermediate. Yeah, and then there will be one right here. That's our 6-carbon intermediate. And then that immediately breaks down into 2 3-carbon molecules. So you have twice as many 3-carbon molecules. So since we started with 3 RUBP and 3 CO2, we made 3 of these 6-carbon intermediates, and therefore we have 6 of these 3-carbon ones now. And then other enzymes come in and continue to modify that. And eventually, what's going to happen is all of these different metabolic processes, all these different enzymes will work on them, and it's going to get reduced. So in the reduction step, we have NADPH being used. Reduction is gaining, right? So now that we've reduced this coenzyme, it becomes a reducing agent. Reduction and oxidation is kind of like playing tag. Once you've been reduced, then you can go reduce something else. So this is where we use up the NADPH that we made in the light-dependent reactions. It's also where we use up some of the ATP in the reduction step. And the ultimate product that comes out of the reduction step is this G3P. Now, we've had 6 of these molecules to be working on. One of them gets to leave. One G3P gets to leave the Calvin cycle, and the other 5 G3P go into the final step, and that's RUBP regeneration. More ATP gets used. I guess I should say in reduction, NADPH becomes NADP plus again because it donates its electrons, it donates its hydrogens. ATP becomes ADP again. Now these two coenzymes, ADP and NADP plus, they can go back to the light-dependent reactions and get recharged. Right? So these two processes are constantly just passing materials back and forth between each other. But we have 5 of these 3-carbon G3P molecules, and we need to use them to regenerate RUBP, and RUBP is a 5-carbon molecule. So we need 3 of these RUBP. 3 times 5 carbons, we need 15 carbons total. Here we have 5 3-carbon molecules, so that's 15 carbons total. So these 5 G3P get converted into 3 RUBP. And that's RUBP regeneration. And then those go right back into carbon fixation. So remember that the enzyme that fixes carbon dioxide is Rubisco. That's the important enzyme you're going to want to know for the Calvin cycle. You're going to know the steps: carbon fixation, reduction, and RUBP regeneration. You're going to want to know that 3 carbon dioxide enter and 1 G3P gets to leave. And if you do this twice, if you use 6 carbon dioxide and get twice as much out, and you get 2 G3P, you can put those together and get yourself a glucose. So what have we seen now? The whole equation of photosynthesis, right? Hold on. Make some space up here. This is the Calvin cycle. Okay. 6 CO2 plus 6 H2O plus light gives us C6H12O6 plus 6O2. The water is split in photolysis, which is part of the light-dependent reactions. The carbon dioxide is reduced to get us some glucose during the light-independent reactions. Okay? So it's not all one big equation. We don't just mix together water and carbon dioxide and get glucose out of it. It's a very, very long stepwise process involving many, many enzymes along the way. So that's overall what photosynthesis is. If you just mix together water and carbon dioxide, you don't get glucose, you get carbonic acid as a result, which is part of the reason why the ocean's becoming more acidy over time, because more CO2 is going into the atmosphere and that dissolves into the ocean as well. Okay, but those are the steps of photosynthesis.